This is Spike the thorny dragon. His spikes are a trait that is coded for by his gene. The genes that code for proteins that can be used in a cell are known as structural genes. However, you may not know that there are also regulatory genes, whose protein products control the expression of other genes by turning them on or off. The regulatory products can affect the transcription of other genes by binding to specific DNA sequences. If the protein is an activator, it will promote the transcription of another gene, and if it is a repressor, it will inhibit the transcription. Gene regulatory networks can arise, which are sequences of regulatory genes whose products essentially go down the line to either promote or repress the transcription of the next gene in the lineup, kind of like dominoes. If the gene is not able to be transcribed, it cannot code for a protein or can't knock down the next domino. Alternatively, if the transcription of a gene is promoted, it can code for a protein, meaning it will knock down the next domino. Let's go back to Spike to see an example. In his cell, gene E codes for the protein that results in spikes being made. The other genes in the network produce regulatory proteins that affect this. S's gene products stimulate the transcription of P, whose gene products inhibit the transcription of I. With I not transcribed, its products are not there to inhibit K, so K is transcribed and its products can stimulate the transcription of E, which gives us spikes in that cell. If we want to look at it like dominoes, S knocks down or promotes P, but then P can't inhibit or knock down I. I can't knock down or inhibit K, but then K is able to knock down or promote E, and we get spikes. If mutations arise in the genes, it can affect the pathway. We will look at some examples. Okay, S is mutated, so P is not transcribed and can't block I. So I is able to inhibit K, but K is mutated anyways and couldn't promote E, so there will be no spikes in that cell. Alright, so P is mutated, so it does not inhibit I. The gene product of I goes on to inhibit K, so K cannot promote E and no spikes are made in that cell. I is mutated, but P inhibits I anyways, so there is no overall change and spikes are made. Now I and E are mutated. S promotes P, who inhibits I, making I's mutation essentially irrelevant. I can't inhibit K, so K promotes E, but E is mutated, so no spikes will be made. Even though all spike skin cells have the same genome, this network still only leads to the formation of spikes in some of them. There are methods in cells that can regulate gene expression without changing the genome. For example, Spike doesn't want spikes on his feet and hands, as this would make things difficult, so the expression of spikes can be regulated in those cells. One type of regulation is DNA methylation, which is where methyl groups are added to nucleotide bases on the DNA strand, often cytosines. Methylation results in the corresponding gene being silenced due to inhibition of transcription. The methyl inhibits proteins from binding that need to bind for transcription. Additionally, the new methylated structure attracts repressive proteins and enzymes that alter the chromatin structure to repress transcription by making it difficult for the players involved to be able to access the regions of DNA that they need to do their job. For example, imagine that gene K is affected by methylation in a skin cell on Spike's foot, resulting in it being silenced. If K is not transcribed, it will not be able to promote E, and there will be no spikes in that cell.